Hello everyone, in this video we'll be discussing about hatch and slag pathway of photosynthesis. This pathway operates in C4 plants and the example of C4 plants are maize and sorghum. So let's start the discussion of this hatch and slag pathway. This pathway is actually a cyclic process. That means the substrate with which we start the cycle would be regenerated at the end of this pathway. Therefore the cycle gets completed and gets on and on repeatedly. This pathway occurs between two types of cells. One is the mesophyll cells and the other one is bundle sheath cells. And bundle sheath cells are the characteristic cells of C4 plants. These are absent in C3 plants. We have dealt with three characteristic features of these cells, bundle sheath cells. The first one was they have large amount of chloroplast in them. The second feature was their wall is thick, which makes gaseous exchange difficult. And the third feature is that they are very compactly packed, therefore they have no intercellular spaces present between them. If someone asks you where are these bundle sheath cells present, the answer is bundle sheath cells are present around the vascular bundle, that means xylem and phloem, in the leaves of C4 plants, and these are present in several layers. So this whole data is from NCERT, so you need to learn it thoroughly. Now let's start the story of hatch and slag pathway. The first half of the story will occur in mesophyll cells. So mesophyll cells are special because they have the primary carbon dioxide acceptor molecule, which is a 3 carbon containing molecule called phosphoenol pyruvate, also called as PEP. So friends, let me brief you what is actually happening. So inside photosynthesis we have a primary carbon dioxide acceptor molecule. So what is the job of this primary carbon dioxide acceptor? This will combine with carbon dioxide and will lead to the formation of a product, and this product is called as primary carbon dioxide fixation product. And on the basis of this primary carbon dioxide fixation product, plants have been divided into two categories. One category is of C4 plants and the other category is of C3 plants. So what is the difference? That in C4 plants the product formed is a 4 carbon containing organic acid, whereas in C3 plants the product formed is a 3 carbon containing organic acid. And here we'll revise the name of these acids. The 4 carbon containing acid formed in C4 plants is oxaloacetic acid, which is also written as OAA. And inside C3 plants, this product formed is 3-phosphoglyceric acid, also written as 3PGA. Now friends, you need to pay attention. Right now we have studied that in C4 plants, this primary carbon dioxide acceptor molecule is a 3 carbon containing molecule whose name is phosphoenol pyruvate, which is written as PEP. So here it is very easy to understand that a 3 carbon containing molecule will combine with 1 carbon molecule of carbon dioxide and will lead to a formation of a 4 carbon containing product. 3 plus 1 is 4. That is easy to understand. But the story has a twist because in C3 plants, this primary carbon dioxide acceptor molecule does not has 2 carbon but has 5 carbon in it, and that is a 5 carbon containing keto sugar whose name is ribulose bisphosphate. So what will happen is that this 5 carbon containing keto sugar ribulose bisphosphate will combine with 1 carbon molecule from 1 carbon atom from carbon dioxide and lead to formation of 2 molecules of 3PGA, which is a 3 carbon containing acid. Therefore we have 6 carbon, 5 plus 1 it's 6, and here we have double the amount of 3 carbon molecules, that is also 6 carbon total. So let me revise it for you that actually what is happening in C4 plants, we have only 1 molecule of 4 carbon containing product being formed whose name is oxaloacetic acid. But in C3 plants we have 2 molecules of 3 carbon containing product formed. The product name is phosphoglyceric acid, also written as PGA. Now coming back to our discussion, that inside mesophyll cells we have the primary carbon dioxide acceptor. We are talking about C4 plants, and whose name is phosphoenol pyruvate. And this phosphoenol pyruvate is present in mesophyll cells, not in bundle sheath cells. You need to compare what is present in mesophyll cells of C4 plants and what is present in bundle sheath cells simultaneously, otherwise you will forget it. So the enzyme which is required for this fixation in C4 plants is phosphoenol pyruvate carboxylase, also called as pepcase enzyme. So here we have dealt with this, what is called as carbon dioxide fixation. So here we need an enzyme. In C4 plants, this enzyme is called as pepcase or phosphoenol pyruvate carboxylase, and also in C3 plants, this enzyme which is used for fixation of carbon dioxide is called as rubisco, and this is the most abundant protein present in the entire biosphere. And what is the most abundant protein found in animal world? The answer is collagen. So these two questions are from biomolecules chapter. So you need to pay attention the enzyme required for carbon dioxide fixation. In C4 plants, it is pepcase enzyme. It is present in the mesophyll cells. And in C3 plants, the enzyme required for carbon dioxide fixation is rubisco. This is also present in mesophyll cells of C3 plants. So now what is happening, that in C4 plants, these mesophyll cells lack rubisco enzyme. So rubisco enzyme is not present in the mesophyll cells of C4 plants, but it is present in the mesophyll cells of C3 plants. The product formed after the primary carbon dioxide fixation in C4 plants is a 4-carbon containing organic acid whose name is oxaloacetic acid, also written as OAA. But you need to see here that oxaloacetic acids has his friends called as malic acid and aspartic acid. These are also 4-carbon containing acids. The name is malic acid, the other one is aspartic acid, and these acids are also formed in mesophyll cells from oxaloacetic acid itself. So now what happens is that we have completed the first half of our story of Hatch and Slack pathway. This is the entire cycle shown in a diagram. The cycle is between two cells. The first cell is mesophyll cells and the second cells are bundle sheath cells. Mesophyll cells are in green color in this diagram and bundle sheath cells are in the gray color in this diagram. So what is happening, we have done the first half, we'll see it first. The atmospheric carbon dioxide would come inside the mesophyll cells in the form of HCO3 negative, that is bicarbonate ion. It will combine with the primary carbon dioxide acceptor molecule, which is a 3-carbon containing molecule whose name is phosphoenol pyruvate. So when carbon dioxide will combine with this phosphoenol pyruvate, it will lead to the fixation of carbon dioxide into a 4-carbon containing acid. The name of that acid is oxaloacetic acid. This oxaloacetic acid has other 4-carbon containing acids as its friend. The name of those acids are malic acid, the names are malic acid, and the other one is aspartic acid. So these two acids have a key role to perform. We'll be dealing it shortly while discussing the role of bundle sheath cells. So till now we have completed the first half of the cycle. Now let's move to the second half of the cycle. How and why bundle sheath cells are so important for this pathway. So inside bundle sheath cells, the friends of oxalic acid, malic acid and aspartic acid, will be transported from mesophyll cells to the bundle sheath cells. Actually, what is the reason that oxalic acid, acetic acid is an enemy of bundle sheath cells? Bundle sheath cells say, oxalic acetic acid dare not come to me. So what happens? Oxalic acetic acid sends its friends malic acid and aspartic acid, which also have 4 carbon each, to the bundle sheath cells for a compromise. But what happens? Bundle sheath cell was really annoyed with this task, and it led to the breakdown of these two acids into a 3-carbon molecule. So what happened? If a 4-carbon molecule releases carbon dioxide, so the product formed will be containing how many carbons? Of course, 3 carbons. So when carbon dioxide is removed from these two acids, which is called as decarboxylation, we get a 3 carbon containing molecule, and ultimately carbon dioxide is released. Now let's see what is happening to this 3 carbon molecule formed. So this 3 carbon molecule formed is transported back to the mesophyll cells, and now these bundle sheath cells, being angry, say to mesophyll cells, dare not send me oxaloacetic acids friend again, otherwise I will lead to their breakdown and will send their dead bodies, this 3 carbon containing molecule, back to you. Mesophyll cell says, chill bro, chill, I'll use these dead bodies to convert into a useful product, which is phosphoenol pyruvate, which is the primary carbon dioxide acceptor in C4 plants. The story begins with this enzyme, this reaction only, where PEP combines with carbon dioxide in the presence of peptidase enzyme and led to the formation of oxaloacetic acid. So when this PEP was regenerated, that led to the completion of the cycle, hatch and slack pathway, and here the cycle gets completed. Now let's see in this diagram what is actually happening. The atmospheric carbon dioxide was fixed into a C4 acid by the action of peptidase enzyme. Now this C4 acid was oxaloacetic acid. It said that I'll send malic acid and aspartic acid, which are my friend, to the bundle sheath cells. But bundle sheath cell got really annoyed and led to the decarboxylation, that means removal of carbon dioxide from them, and the dead body had C3 carbon containing acid. And this 3 carbon containing acid was transported back to mesophyll cell. But mesophyll cell was smart enough to convert this C3 acid again back to phosphoenol pyruvate, which will again combine with the atmospheric carbon dioxide in the form of bicarbonate ion, and the cycle will continue on and on. Now you need to pay attention to the fact that during the process of decarboxylation inside bundle sheath cells, there is release of carbon dioxide. Therefore, if I ask you in which cell, mesophyll cells or bundle sheath cells, the amount of carbon dioxide will increase, the answer is in bundle sheath cells the amount of carbon dioxide will increase. You need to remember this point. Now what will happen to this carbon dioxide? This carbon dioxide will enter a cycle called as Kelvin cycle. And friends, Kelvin cycle will occur in all the plants, be it C4 plants or C3 plants or say CAM plants. If it is a plant, then for sure Kelvin cycle will occur, because Kelvin cycle is the key cycle which lead to the formation of glucose in plants. Understood? Now let's discuss what is the fate of this carbon dioxide released inside the bundle sheath cells. So this carbon dioxide released inside the bundle sheath cells will enter Kelvin cycle, and bundle sheath cells are rich in the enzyme rubisco. Now you need to pay attention that Kelvin cycle has 3 steps. The first step is that of carboxylation, and inside this first step what happens, that carbon dioxide molecules will fix with RUBP, which was a 5 carbon containing keto sugar, and will lead to the formation of 2 molecules and will lead to the formation of phosphoglyceric acid, that too 2 molecules. This is a 3 carbon containing acid. And my dear friends, you should remember that the story in C3 plants directly starts from this reaction. The carbon dioxide fixation in C3 plants is directly done by RUBP, which is a 5 carbon containing keto sugar. But inside these C4 plants, they have a long loop. What is actually happening? This carbon dioxide will be fixing with PEP, that is phosphoenol pyruvate, inside in the presence of peptidase enzyme, and will lead to formation of oxaloacetic acid. This oxaloacetic acid will convert into malic acid and aspartic acid. These 2 acids will be transported to bundle sheath cells. There they will go decarboxylation. Their carbon dioxide will be released, and that carbon dioxide will enter this first reaction of Kelvin cycle. Therefore, they take a long loop. But there is an advantage of this, that by doing this long loop inside C4 plants, what happens is that in bundle sheath cells where the rubisco enzyme is present for the C4 plant, the amount of carbon dioxide in the surrounding increases, and this lead to the important function that this enzyme will function only as carboxylase. Later on, when you will see the video of photo respiration, there you will understand what was the importance of this carbon dioxide being increased at the level of this enzyme Rubisco, because this enzyme has no character. It just depends upon the surrounding people. If the surrounding peoples are more in carbon dioxide, it will react as carboxylase enzyme. But if the surrounding has more of oxygen people, then this enzyme would react like oxygenase. It is just like a mirror. So friends, this was the first step of Calvin cycle. Second step is of reduction, where this product formed phosphoglyceric acid would be reduced in a series of reaction to triose phosphate, and this triose phosphate, some part of it would be sent to the formation of glucose and some would be sent back for the regeneration of this 5 carbon containing keto sugar ribulose bisphosphate. So you need to pay attention that Rubisco enzyme in C3 plants is present inside the mesophyll cells itself, but in C4 plants it is present inside the bundle sheath cells. The second thing is that in mesophyll cells of C4 plants, Rubisco is absent, but pepcase is present. Pepcase was the enzyme required for the carbon dioxide fixation inside C4 plants, which led to the formation of oxaloacetic acid, that reaction. But this pepcase enzyme is absent in bundle sheath cells. With this, friends, we have completed the thorough discussion of Hatch and Slack pathway. What was the importance of taking a long route to formation of carbon dioxide? Just because by taking a long reactions, the amount of carbon dioxide at the reaction site was increased, and therefore Rubisco enzyme functions only as carboxylase enzyme, not as oxygenase. With this, the discussion is over. Thank you for watching this video till the end. If you liked my content, do check my other videos also. And friends, I share my notes on my Telegram channel. Do visit my Telegram channel also. Thank you for watching this video.